We're done talking about your monosaccharides, so let's go to your disaccharides. Your disaccharides are formed by the fusion of two monosaccharide units via what is called a glycosidic bond. So we see here the process of condensation. Let's say we have alpha D glucose and beta D glucose. So the process of the formation of your glycosidic bond is again a consequence of what we learned earlier. So the consequence of a hemiacetal to condense with an alcohol functional group. So recall that carbon 1 in your monosaccharides forms a hemiacetal. So this is a hemiacetal, and this can condense with another alcohol group. But your glycosidic bond is formed if the alcohol group comes from another monosaccharide. So in the process of condensation, one water molecule is released and you form this O-glycosidic bond. So O there means that the two units are formed, or the two are bonded via an oxygen atom, because we also have what is called the N-glycosidic bond, which you already saw when we discussed your nucleic acids. So the N-glycosidic bond, you have the nitrogenous base attached to your ribose or deoxyribose. So their attachment is via a nitrogen atom instead of an oxygen atom. So that is your N-glycosidic bond. So in naming your disaccharides, we have their common names such as maltose, which is formed from the condensation of two glucose units. But we also have a systematic name. So what you do is first you start from the left unit or the leftmost unit, and you indicate its anomer. Is it the alpha or is it the beta anomer? And then is it the D or the L isomer? And then the name of the monosaccharide. And you can include there whether it is the pyranose or the furanose form if you want to lessen ambiguity. Dash. Then we enclose in parentheses the numbers of the carbons involved in the glycosidic bond. So for maltose, in the leftmost unit, we have carbon 1. So we put there 1, followed by an arrow pointing to carbon 4, which is the carbon involved in the glycosidic bond in the second glucose unit. And then we indicate whether the second, so we are now in the second unit, whether it is, if it is in a different anomer, if it is beta, then you should put there beta. But sometimes, if they are the same alpha anomers, there's no need to place their alpha. And then you put, is it the D or the L isomer, and then the name of the monosaccharide. Of course, it's easy if we are talking about disaccharides, but what if we have a tetrasaccharide or an oligosaccharide composed of 10 monosaccharide units? It would generate a very long name if we follow these systematic rules. So what they do is they designate abbreviations for the common monosaccharides. So these are three-letter abbreviations such as GLC for glucose, GAL for galactose, etc. And they use this to shorten the systematic name. So for example, maltose will have such shortened systematic name. So you have their GLC for the glucose units, and you have alpha indicating it is the alpha anomers involved, and you have the linkage there, carbon 1 in the left glucose unit and carbon 4 in the next unit. Your hemiacetals, so that's your carbon 1 in your monosaccharides. Hemiacetals can actually reduce other substances. So that carbon 1 in your monosaccharides, those are reducing agents. So because it's possible for them to be oxidized, so they are reducing agents. So all of your monosaccharides, since they have the carbon 1 available and it's in the hemiacetal form, they are reducing agents. And this is the basis of one of your qualitative tests. When it comes to disaccharides, it's possible for a disaccharide to lose its um, ability to reduce other substances. Okay, so we call that the reducing end. So the carbon 1, the hemiacetal, is called the reducing end. And in forming disaccharides or other oligosaccharides, it's possible that the carbon 1 in the rightmost end of your oligosaccharide be involved in a glycosidic bond. And the consequence is it's no longer available to reduce another substance. So we lose the reducing end if the rightmost or the last one, the last unit, in the last unit of your oligosaccharide is its carbon 1 is involved in the glycosidic bond. So an example, let's take an example, sucrose. So in sucrose, the units are glucopyranose and fructofuranose. So those are the units. And the linkage is, so you have the alpha, or so you have alpha glucose, carbon 1 from glucose, and carbon 2 from fructose. Now, these are the anomeric carbons of glucose and fructose respectively. So carbon 1 for glucose and carbon 2 for fructose. Since these two carbons, which are the reducing ends of the corresponding monosaccharide units, are involved in the glycosidic bond, so they are now an acetal, right? So because we lost the OH functional group in the formation of the glycosidic bond. They are both acetals now. So carbon 1 in glucose, this is an acetal. Carbon 2 in fructose, this is an acetal. Since they are now acetals, they can no longer be reducing agents. So in the formation of your glycosidic bonds, it's possible that the reducing end becomes involved and we lose the reducing end. So they will test negative in that qualitative test I mentioned. And if you note here in this reference, in the name, they use double-headed arrows. So some references would use double-headed arrows to emphasize that the hemiacetal carbons are the ones involved in the glycosidic bonds. And then we go to polysaccharides. So your polysaccharides are made up of 20 or more monosaccharide units, and we can classify them into two major groups. We have the homopolysaccharides and the heteropolysaccharides. So from the name itself, this means the same. So your homopolysaccharides are those made up of only one type of monosaccharide. An example would be starch. Starch is composed of only glucose units linked together. Glycogen is also a homopolysaccharide. Like starch, it's also made up of glucose. So their difference is their composition. So glycogen is made up of glucose units attached 1 to 4. And then there are branches every 8 to 12 residues. So a branch is created via 1 to 6 linkage, and that's your glycogen. Starch, on the other hand, is made up of two types of glucose polysaccharides. We have the amylose, which is completely linear, glucose units linked 1 to 4, completely linear. And we have amylopectin, which is like your glycogen. The difference is the frequency of the residues. So there is less frequency of residues in your amylopectin. So the branches, which is the same, 1 to 6 linkage, occurs every 24 to 30 residues. So they are different because of the composition of the polysaccharide. Now, heteropolysaccharides, an example, are those polysaccharides attached to your proteins involved in cellular adhesion and recognition. In the extracellular matrix, some of these heteropolysaccharides actually provide the framework where your cells are attached. Your agarose, so we have encountered agarose in the previous module. In agarose gel electrophoresis, it's also a polysaccharide. It's a heteropolysaccharide. It's made up of D-galactose with 3,6-anhydro-L-galactose, and so on. So your polysaccharides have a wide variety of functions. Starch and glycogen, they are storage of energy, right? Cellulose, chitin, these are for structural support in plants and your crustaceans. Peptidoglycans are also structural polysaccharides. They are part of your bacterial cell wall. Hyaluronan, a glycosaminoglycan. So from the name itself, it's composed of carbohydrates and proteins. So this is also structural, found in vertebrates. So we've seen that aside from the types of monosaccharides that you can put together through your glycosidic bonds, the same units can link in different ways. So we've seen that there's the 1,4 linkage, 1,2 linkage is possible, 1,3, 1,6. So this adds another dimension of variety to your polysaccharides. And I think this level of variety is higher than that of what we've seen in your proteins and your nucleic acids.